All right, good morning, everybody. I think we're going to go ahead and get started. Good morning again, and welcome to the Acres USA webinar series. I'm Ryan Slaybaugh, the Executive Director here at Acres USA, and I'm the host of this series. We're recording live on July 26, 2022. This will be recorded and available on our YouTube channels very soon. Generally, we try to get it up the first 24, 36 hours after the event, so we'll make sure that link is available and out there. This is also available for CEU credits, so please find those details in the chat. Thank you, Hannah Fields, for producing this and posting those out there today as part of our team. And if this is the first time at one of our virtual events, you can learn more about Acres USA at acresusa.com. Today's webinar is sponsored by Holganix, a company on the cutting edge of the input industry. They're researching and producing microbial-based products that can be used in the spring, in the fall. They're working on one that can be used around harvest right now, so the timing is very good today to learn a bit about the roles microbes play in soil health and crop protection. So today we'll be talking about the role soil microbes again are going to be playing. We're joined today by Dave Stark, the President of Agriculture for Holganix. He's a scientist by trade, and today he's going to put on his teaching hat for us. So we'll follow his 30-minute talk with a few minutes of Q&A. So if you have questions, please drop them in the chat. We'll collect them along the way and make sure that they get answered at the very end with about 10 or 15 minutes, depending on how many questions you have. So again, thanks everybody for joining us. Sounds like from around the country and many in Portugal even today. So we're proud to have everybody on board for the talk today. So go ahead, Dave, take it away, and thank you for joining us.
Okay, thanks, Ryan, and thanks everyone for taking the time to spend with us. We're going to talk about soil microbes. So again, as Ryan said, I'm a scientist by training, a molecular biologist of all things. I spent almost 40 years now in agriculture, a big ag company doing a lot of things from science and business unit leadership to Holganix. One of the things I've seen through my career is how much new technology gets thrown at you guys, and how you keep it straight is beyond me, because I struggle. And the area that Holganix is in with soil microbes is one of them. So I'm not here to sell you. I'm not a salesman. I'm here to help explain what to look for in a soil microbial product so that you can pick out the products that match your expectations and your needs. Because there's a lot of good products out there, and there are some that probably don't do everything that they claim. So really, like Ryan said, I want to have my teaching hat on. So one key thing, how we build soil is through microbes. Microbes are what makes the soil work. So we want microbial activity. I think on the surface, all of you know that. When we don't have good microbial activity, that's how we get, soil that is, rock hard and doesn't hold water and has no nutrients in it, because the microbes will eventually destroy the soil if they don't have plants that are working with them to feed each other. So, I'd say microbes, and I know just through the years, I get a lot of questions and such. So first, we're going to start off, what do I mean by microbes? So these are the different types of soil microbes. And by the way, microbe, what do I mean? Scientists are real clever. It means they're so tiny, you need to use a microscope to see them. So they're microbes or microorganisms. The most abundant, the smallest, the fastest to grow are on top. They're the bacteria. And as you go down the list, there are actinomycetes, fungi, protozoa, etc. They get larger, a little less numerous, and slower to grow. All of them are needed to have healthy soil function. This is a precision, high-tuned engine. And if you pull components out of an engine, it doesn't work very well. It's the same with the soil. So, one key point, if you look at a teaspoon of healthy soil, there will be over 1 billion of these individuals. There are thousands of species. Actually, there's so many species, scientists, we feel that we haven't identified even half of them yet. So the soil is very alive. So let's just start with bacteria. Bacteria are important. They're some of the workhorses in the soil. They're good at breaking down organic matter, crop residue, manure, whatever. They unlock nutrients in the soil. They induce rooting. So if we remember, I said microbes and plants work together. Well, plants will spend 30% of all their photosynthetic energy, a massive investment, just to secrete food in the root zone to attract microbes. So 30% of their energy. Why? Because microbes access food in the soil the plant can't. So the roots are the home of microbes, not just bacteria, but everything I showed. And so if you have a home that feeds you, a lot of times you want a bigger home. So there are microbes that secrete hormones that induce rooting. You'll see microbial products out there that talk about the roots they induce. That's pretty credible. That's what microbes will often do. Now with bacteria, even though there's a lot of good things, there's some things that aren't so good with a bacteria-only product. And I should point out, most of the products that you're being sold or you're being promoted to you only contain bacteria. They're cheap to grow, they're easy to stabilize. They're important, but they're not sufficient. So if you want to put carbon in your ground, bacteria tend to release it as CO2. They dominate most farm ground anyhow, because we till, and tillage encourages bacteria growth, not the other things. And we're going to talk a lot about carbon to nitrogen ratio. And I'm sorry, because this is going to be a little bit technical, but it's so important to everything I'm saying about why you need biological diversity. So bacteria have a very high nitrogen requirement. For every nitrogen atom, they have 5 carbons. So if the only thing you have in your soil are bacteria, they're going to tend to tie nitrogen up because they're nitrogen hogs. So now let's look at the next class we'll talk about, fungi. So fungi are also really good degraders at breaking down organic material. Actually, they're better than bacteria because the woody residue, not just trees, but I mean the stuff that sits in your field a long time that's slow to break down, like corn stover, wheat straw, barley straw, those are very woody. They have components we'll talk about later that really only fungi produce the most efficient enzymes to degrade. So we want bacteria, we want fungi in the soil. They also consume bacteria, help keeping them in check. They're really good at finding water and nutrients, at unlocking things like phosphorus and potassium in the soil. And you look at the picture, look at all that white fuzzy material. That's mycelia, that's fungal hyphae that group together to form that white stuff that go all through the soil. They get between soil particles, even your plant, hair on the roots can't get into. That's carbon rich. So unlike bacteria, fungi do a better job putting carbon in the soil. Carbon, of course, whether we get paid for or not someday aside, it helps your soil work better because it's food for microbes and adds soil structure. Fungi require oxygen. So if you flood it out, you tend to lose your fungi. They're also knocked back by tillage. Now I'm going back to my carbon-nitrogen ratio. Remember, 5 carbons, 1 nitrogen in bacteria. Fungi, and there's a range, on average 3 times less, 1 nitrogen for 15 carbons. So they don't tie up nitrogen as much as bacteria. And to me, sadly, most farmland needs more fungi. Most microbial products don't contain fungi, or they can contain 1 or a few. And then the last one I want to talk about are protists, and you probably don't hear about these at all. The amoeba, protozoa, these are things that move around in the soil, and they're great predators. They eat bacteria. A single amoeba or protozoa can eat 10,000 bacteria a day. So they really help balance out. Because what I didn't mention is, bacteria, while they are the fastest to grow in the soil, the fastest to replicate, and in an ideal growth conditions, bacteria can double every 20 minutes. They will get the first bite of any nutrient in your soil. Your fertilizer, manure, whatever you put out there, bacteria get to it the first because they're the most aggressive and most numerous. So that's good because remember, all these microbes I'm talking about live within 1/2 inch of a root. So they immobilize the nutrients in the root zone. but we need the fungi and the protists to get the nutrients out of the bacteria and back to your crop. So when we took a look at that carbon-nitrogen ratio, remember, bacteria 5-to-1, fungi 15-to-1, protists are like you and me, 30-to-1. So as they eat their 10,000 bacteria a day, they have to get rid of the nitrogen. It's toxic to them. And it's just an amazing way that God designed all this, that the bacteria are the most aggressive. They grab the nitrogen, the predators release it. It all happens in the root zone. And what do protists release? Ammonia and nitrate. What are the two forms of nitrogen your plant root takes up the most? Ammonia and nitrate. None of this is an accident. And so now, just as an aside, as a molecular biologist, our specialty is DNA and genetics. And I know all the evolutionary theories. And I'm kind of passionate about this because I bought into them. And I've got a lot of people that I hear talk about millions and millions of years of evolution have perfected this. I got to tell you, looking at the data, coming from a place where I used to buy into it, I was wrong, and I'm ashamed. This is not millions of years of evolution. When Jesus said from the time of creation that he created these things, that's actually what the data supports, that this is all designed. Mathematically, for any of this to come about randomly, the mathematical term is absurdly impossible. So if you've gone through life, believing in Genesis chapter 1 as a scientist, the data is on your side. Just thought you got to know that. All right. So again, protists, and by the way, very few microbial products have protists. These things are important. Now, I get asked all the time, if I use your product, do I need to keep using it every year? I've got good soil. Microbial populations are in constant flux. And there are things that can really disrupt the balance. Because remember, we need everything on that list to keep cycling nutrients so things don't get tied up or lost. And we build topsoil. So tilling, like I said, we promote bacteria. Overuse of some chemicals, certainly a soil fumigant kills everything. Overuse of fertilizer tells the plant you got enough nutrients, don't invest 30% feeding the microbes. Well, that disrupts the microbes. Those are things we can control. There's things we can't. Flooding, 48 hours underwater, we start losing oxygen in the soil. There are certain types of bacteria that can survive with or without oxygen. Yeast, which is only one, the fungi doesn't need oxygen, but all the other beneficial fungi, which do everything I just said, need oxygen. And of course, so do all the protists. So you can tell if your soil's way out of whack, when that water drains on that field, it smells like the bottom of a swamp. That's bad news. Now your biology is way out of whack where you just have certain types of bacteria. Freezing also kills bacteria. They're full of water. So adding a diverse microbial back can help bring that balance back to the soil. So remember, we want to cycle nutrients, and we need all pieces to keep them from getting tied up. And we're going to talk more about, how the microbes help with residue and such going here in a few slides. Another thing that, I talked about roots and a lot of microbial products, how they promote rooting. And, I've given a number of talks about how microbes help with fertilizer efficiency. And fertilizer, the way we use it, is really inefficient. There's a lot of science that says less than half of what we put on the ground goes to the crop. So if we spend, in 2020, over $18 billion in fertilizer just in the United States, that means that we're all wasting over $9 billion losing our fertilizer to the environment. So what do we do to help it better? Get more bang for our buck? Roots is one. So a typical root zone, the roots only explore 1% of the available soil, 1%. And many nutrients need to come in contact with the root to be taken up. So if you're using a product that gives you more roots, You're going to find more nutrients in the soil. You're going to get better fertilizer efficiency. Another aspect, though, that's also even more important, I think, is those roots. They break up compaction layers. They make channels for air and water. And when they die and degrade, they become the stable organic matter in our soil. So there's a lot of science that shows 2/3 of that carbon in our ground, the organic matter that gives us good, rich soil, actually comes from roots. It's not the residue left on top. So roots matter. This is just, again, another slide. I talked about fertilizer efficiency. I'm not going to spend a lot of time here on it, but just know there is a massive body of scientific evidence that shows that the more biologically active your soil is, the higher your yields will be per unit of fertilizer input. So the more biological activity you have, the more bang you're going to get out of your fertilizer dollar. So we want to do things that encourage diverse microbes, not just bacteria, because they can tie it up. We need all the different species. This isn't just nitrogen. This is also phosphorus and potassium. So in the top 6 inches of soil, and this is an average, so your ground could be different, but there are hundreds of pounds to maybe over a ton of phosphorus, and there are tons of potassium already in your ground. And this is typical of almost all the ground that we farm. However, the available pool, so when you do a soil test, you get available pool, is tiny, less than a pound of phosphorus, a few pounds of potassium. It's all there, but it's in a chemical form the plant can't take up, but microbes can. This is one of the reasons plants feed microbes, to unlock P and K. So now let's look at what microbes do for crop residue. So, residue management, I know there's value in the residue as an animal feed or an energy source. There's a ton of value in your soil. So, for example, in corn, for every 40 bushels roughly, you're producing a ton of crop residue. So a 200 bushel an acre corn crop can have 5 tons of residue on it. Each ton has 17 pounds of nitrogen, 4 pounds of phosphorus, 34 pounds of potassium. And potassium for stalk and stem strength is so important because there is so much. 3 pounds of sulfur. There's a lot of value in that residue. Not only that, the carbon in it, the food source, the organic matter, is food for the microbes, and we want to encourage microbes. But here's the balance, because we also know we need animal feed. So a lot of times we want to remove some of that residue, but if we remove too much, then we get erosion, and erosion is awful. You lose your topsoil, nutrients, and frankly, a lot of your microbes. You can get crusting, of course, when it rains, and we decrease the quality of our soil because we're not replenishing the organic matter. Too much residue is also a problem. It can jam the planter, it can make getting good seed placement a problem, and also it can really lead to uneven warming of soils. The soils will warm slower, so you can't plant as early, but will warm unevenly. And especially in a crop like corn, uneven emergence can cost a lot of money in terms of yield at the end of the season. So we want to manage, find this balance. So what can we do to manage the decomposition rates of our residue? Well, it's all microbial activity. The only thing that breaks down your residue are microbes. So the better question is, what are the factors that influence microbial activity? Number one, you got to have the right mix, bacteria, fungi, protists out there, especially fungi, because fungi have the biochemical capability of breaking down that hard to degrade residue. So if you're looking at products to help you break down your residue, you need fungi. The other main factors, and there's a lot here listed, are going to be moisture and temperature. So if it's cold or if it's bone dry, the microbes aren't going to do much. If it's warm and moist, they're going to work really well unless they're not there to begin with. So any product you buy to break down residue is going to depend on temperature and moisture. So I love this slide. I have a point, so don't worry, there's no test. What is it with our residue that makes it so hard to break down? Well, there's three main components, and they're polymers. So they're like a sugar that's linked together that becomes a polymer. There are three main polymers. These are the most abundant polymers on the planet: cellulose, lignin, and hemicellulose. That's almost all of the dry weight of our crop residue. Cellulose is a straight chain, and it becomes these rods. I don't know if you can see them there in the middle, the microfibril. They're very rigid. You can pack them close together. They, along with the lignin, which is a much more amorphous molecule, kind of an iron shield that keeps those cellulose microfibrils together, that's what gives, a 200-foot tall tree that weighs tons and tons the strength to stand up to 100-mile-an-hour wind. That's what gives the crop, like your corn stalks, the ability to stand up. They're very, very strong. Hemicellulose is kind of a glue. Now there's a point. Remember, I've been talking about carbon-nitrogen ratios. Look at the chemical structures. C is carbon, O is oxygen, H is hydrogen. All the little lines that are connecting, we're lazy as scientists. Those are all carbons. Those are just carbon-carbon bonds. We just draw lines instead of C's. Where's the N? There isn't any. There's no nitrogen. What's the carbon-nitrogen ratio of the food stuff that only microbes can break down? It's terrible. So we have to have nitrogen. So for example, here's a chart showing that some of the residue, like alfalfa or from legumes, even soybeans, unless you've got a big bushy crop, it breaks down pretty fast. Those have more nitrogen. So the microbes can do the job and actually release the nutrients. But you get into corn stover, wheat straw, where there's so little nitrogen, it's harder to break down. So if you use especially a bacteria-only product, it's going to take longer to break it down because they don't have the right enzymes. They're also going to steal your nitrogen, because to break down that residue, they need to have nitrogen, and it's not in their food, it's in your soil. So you can actually see in the spring where you've put nitrogen down, corn that looks like it's nitrogen deficient, because for a period of time it's tied up. So what you want is a product that has all of the microbes, and whether this is for fertilizer efficiency, building soil health, breaking down residue, our product is one of these. So we call it Bio 800 Plus, and we have a couple. One, agriculture for spring planting to induce rooting and get more bang out of your fertilizer. Break down residue. All of them contain over 800 species, over 600 bacteria species, over 200 species of fungi, over 70 species of protozoa, plus food sources. How can you tell if what you're using has all this? Well, it's really the form you buy it in. A lot of microbial products come as a powder, or they come in a liquid in a tightly sealed jug you can keep on the shelf at room temperature for months. You cannot stabilize protozoa that way. You can't stabilize most fungi or even a lot of bacteria that way. You can stabilize spores and spore formers. That's a narrow class of bacteria and a few type of fungi. So if you're buying a product like that, that comes in a form like that, then it's probably narrow biology. Our product, because these are living microbes, the only way we stabilize it is refrigeration. We have to keep it cold, just like you refrigerate your yogurt for our biome in our guts. We refrigerate our product. As an end user, you don't need refrigeration. You've got weeks to work through it, but we, when we make it and store it, do. So these are kind of some of the clues to look for. And now it gets salesy, and I'm not going to do that because I'm a scientist. I just want to show you that microbes are a tool if you've got diverse microbes. And by the way, compost teas and products like that also can be diverse. So there are some things out there. They're tools to help you reduce fertilizer. On our website, we have all kinds of data on this. These are 2 years of studies in Illinois showing that you can reduce fertilizer, or you can keep it the same and get higher yields. It's really a tool in your operation to help you pencil out what works best for you. I can tell you, I've got customers after using our product for 3-4 years, they started at maybe 1 or 1.2 pounds of nitrogen per target bushel of corn that they wanted to harvest. They're at half of that, they're at 0.6, and they're still doing north of 200 bushel an acre corn. So again, it's a tool. I'm not saying you can do that because your operation and your soil is different, but it's a tool that will help you be more efficient and make more money. This is Pasture. This is just reseeded in fescue. This is the same pasture, and one part they used the microbes and one they didn't. No fertilizer. You know, on the bottom you can see the microbes are releasing nutrients and feeding the plant, helping the root zone versus having no microbes. Same in corn, higher yield. And I said, you know, before, potassium is important for stalk strength. There's a lot of microbes that will unlock the tons of potassium you've got already in your field and feed it to the plant. And here's just some evidence of it. And again, a lot of products out there can do that, not just mine. Microbes are good at that. Data on wheat and yield, again, it's get salesy, Tennessee. I don't want to do salesy stuff. That's all on my website. The point is, diverse biology really does help you produce more using fewer inputs. And I would just say that use it as a tool to help find the sweet spot in your operation. This is a picture of residue breakdown. So this is from Central Ohio. And here he didn't apply, because most residue breakdown products should be applied in the fall right after harvest. But if you're in a place where it's winter after harvest because you had a wet fall, then you don't want to put a microbe down. It's too cold. So here he put it down with this herbicide burn down. And by the time, you know, he planted his beans through it, he said he was already amazed at how much of that residue was already breaking down. Now, this isn't a crop, this is turf. This is wheat straw. You can see without the microbes, that straw after 7 months has hardly done anything. So is the grass too. With the microbes, the straw is almost gone. And of course, the grass is so much more full because the microbes are inducing rooting and putting nutrients back into the crop. That's what they do when you have the diverse biology in the soil. You know, another advantage of using a breakdown product that's got diverse biology, you apply it in the fall after harvest. And if you're going to, like here, drill rye as a cover crop through it, or if you're going to do winter wheat or any other cover crop, the microbes will help, just like in the turf, with the rooting and the establishment. And, you know, often between harvest and getting the cover crop established is a race to winter. What happens first? So any edge you get to get that cover crop up and established while getting a head start on breaking down the residue is probably good for your operation. So I threw a lot at you, and I apologize. I'm glad this is recorded. So let me distill it down into a few key points, the takeaways. Microbes are what make the soil work, help you get more out of your fertilizer, actually builds the quality of your soil and the value of your farm, because you've got better soil that will produce higher yields. But it's diversity. If you want a microbial product that does all of this, you need to have one that has bacteria, fungi, and protists, not just bacteria. Those products could be fine if your only problem, for example, is unlocking phosphorus, or if you're trying to target a specific disease or pest. They can be very effective. But to overall help your plant health and soil health, you must have the diversity. And I think you'll find that it's a really valuable tool. And you should just kind of play with it on your farm and see how it works. That's my email address. If you've got questions, email me. Feel free. And I thought it best if I just end with a quote from, sadly, the late Dr. Fitzpatrick from Acres U.S.A. magazine, talking about the importance of diversity. So with that, again, I'll take your questions, and I appreciate your attention. Thank you very much.
Thank you, Dave. I really appreciate the talk. You got through a lot in a little bit of time, which is no small feat. So I appreciate that. It's a big topic these days and rapidly developing an interest. You can tell from our chat, and the questions that we have coming is large. So I'm going to get right into it to make sure we have time for questions, about a minute a question or so, just to kind of give you a parameter if we can work through, just to get through them all overall. We'll start with one Ann asked early on in the talk. When you say your control roots have some that are longer, does that mean that you're seeing them access deeper water reservoirs?
I'm sorry, I missed. What is longer?
The control roots that you're I guess the root systems that you're seeing in your test.
Oh, sure. So, I mean, with a lot of microbial products, including our own, the difference in the root mass is an easy way for growers to see that they're getting a response. And absolutely, they go much deeper and they access that water that's further in the ground. So it's not just at the surface where it dries out faster. And remember, the root hairs, and that's something I didn't mention, some fungi like mycorrhizae are so tiny, they find water in soil particles that are too small for the root hairs. So they really work together for water too.
Great. Well, thank you. We'll ask another question. This one's from Gladys. What is an accept? And this is kind of related to another question, actually. So maybe we can talk about just measuring microbial life in the soil. I know that's was one of Dr. Fitzpatrick's specialties, as well as to bring it full circle. But what is an acceptable and high microbial activity level? And then how are you measuring it to make sure you're getting consistent data?
You know, that's a phenomenal question. And when I started, with Holganix, I was eager to talk with soil scientists. And that's one of the questions I asked. And you know what they always said? They'd put their head back and laugh and say, yeah, I got the same question too. That's not a very good answer, but it's an honest one. Microbial populations are in constant flux. They're influenced not only by what you've got growing out there now, but by what you had last year. Because even varieties of the same plant will attract slightly different microbes. So the truth is, we don't know which of the species that you need to be ideal. Science hasn't caught up. We do know the functionality we need, which are the different main types of microbes. And we do know that if you measure total microbial activity, but also look at things like the fungal population or the bacteria to fungi ratio, then you're getting an idea. If you're just looking at oxidative burst, that could be from a handful of bacteria. You have a lot of activity, but from what you just heard from me, you don't want just bacteria. So you need to go deeper. But spending the money to do like a DNA fingerprint, which we do on all of our products, so I can tell you every species, I don't think we're there yet. I wouldn't do it.
Makes sense. If you are in measuring, where are you measuring for the soil, for the biology?
So we're doing some studies now, and we're going to measure, the key is soil moisture is such a big deal for microbial activity. So if you're going to sample soil, you've got to make sure it's as uniform as you can. So you want to take it certainly within the top 6 inches, and you want the same moisture levels. And that's what we're doing in the root zone, because that's where the action is. So we actually will dig up plants and take some of that root, that soil. Depending on the test you use, they're going to tell you, what the best samples are. So they're pretty good about that. They know their strengths and weaknesses.
Makes sense. I appreciate that. Related, somebody asked about in a non-drought environment, in fact, the opposite, in a heavy moisture environment. Are you seeing the same, results with your product, or are they as effective in heavy moisture environments?
They are, with the caveat that, again, if you're under a flood situation, that's going to knock back a lot of the beneficials. And again, it's an extended flood. And so I lived 35 years in St. Louis, Missouri, and it seems in the spring, the rivers, especially the Missouri and some of the tributaries, always flooded. And I like to run at some of the parks, and you could smell it before you saw it. And that's a problem. So when that happens, you want to add the biology back after the flood water is receded to give it an instant start. Will it recover on its own? Slowly, but it will. But if you're trying to grow a crop now, you want to add the biology back.
Makes sense. I appreciate that. Question related to product mixing. Ultimately, you mentioned that a couple of your clients were using, herbicides or more conventional herbicides with your product overall. Is that a cost prohibitive practice does the chemical interfere with the biology in your product?
I know a lot of microbial products don't want you to tank mix with anything. They even want you to use dechlorinated water. And if the only thing I was concerned about was how perfect my product could be to give me every advantage to win, I would make the same demands. But that's not practical. So we have years of experience. You can tank mix with anything. Use the city water. Just don't tank mix with fungicide. Because again, that's one of the key components we're trying to add. You can have fungicide on the seed coat. That's okay. But just don't tank mix with fungicide, and then you're good to go.
Appreciate that. We'll stay maybe on the product specific things because we got a few questions on that. One related to the research that you've done behind this. What kind of replication experimentation is going on to replicate the research that you're finding out there? And then what's your standard error? I love our audience sometimes and the question you're asking. So thank you for the thoughtful questions.
Yeah, so you'd have to go to our website. We have all kinds of studies. And I'll tell you something, generally speaking, there are certain groups that like to do short replicated rows in trials, like 20-foot rows. And the standard error in that is massive. So I've got a bunch of studies, and these are all very good researchers and really good land grant universities, where the standard error can be 100 bushels between different reps. So what I like are larger scale trials where it's split field, or you're taking half the planter, as the treatment, the other half is the check, and you just go up and down. So you're correcting for topography, soil type. Even a uniform field, believe me, is not uniform. So we do all of that. And I can tell you my 26 years at Monsanto is, measuring yield is why I'm thin and gray, because it's so many variables go into it.
It's a good point. Something we don't always encourage on a short-term basis, of that type of measurement. It can send you down some wild goose chases if that's your only measuring stick. Yeah, absolutely. Related, we're going to get to a couple more product questions, and then we'll get back to a couple of soil health questions that we have out there as well. So I appreciate those patients who aren't getting their, those who are patient waiting their question to get answered here. Next question would be, how do you determine the culturability of the mix of microbes in your product? Do you follow DNA fingerprinting, or do you have a viability assay?
No, we do DNA fingerprinting. So we have a process that's very deliberate through our. So we have a stock inoculum, so we know what we're putting in the diverse biology, but we also know the different families and types of microbes need specific conditions. So we change our conditions through our manufacturing process to stimulate each component we want, through different things that we can control in that process. And at the end, we DNA fingerprint. So the DNA fingerprint, I can't put on the label because it's just too big. But if you email me, I'll send you a sample. There's no secret.
Appreciate that. Absolutely. Again, you can learn more at his website at holganix.com as well. We got that in the chat room if you want to dig into the research behind all this. We're going to get back to a couple soil health questions generally, and to kind of round out the talk today. One's from Frank, and we appreciate that. His question is this true that if you have very healthy soil, then you have very healthy fruit, and insects prefer to eat unhealthy fruit? Is that generally true?
It's generally true. And we see that, with our product and with others. And it's counterintuitive being a human being and a father of daughters who I remember when they were little loved sugar, that insects are actually opposite, that high sugar repels piercing and sucking insects. So white flies, aphids, and we see this time and again, that they go after the weaker plants. And as a scientist, all I can surmise is because those plants are less able to launch their full defense response. So they're more susceptible. And I'm sure I know there's tons of science around this, but the short answer is, yeah, you're right.
It's one of the truths that truly are giving these type of products and this line of research a lot of acceleration, a lot of fuel right now. So appreciate that. A couple more questions here. Can you talk about the relationship between amino acids and the microbial life? Secondarily, would it be beneficial to add amino acids to help feed your microbes?
Yeah, we actually put amino acids from yeast extract in our mix because they need the nitrogen source, and amino acids are phenomenal sources of nitrogen for microbes. A lesser extent to the plants, a few amino acids plant roots can take up. So sure, I mean, and we put a pound of molasses in there because they need the sugar, and also some micronutrients, copper, magnesium, iron, et cetera. None of that supplements your soil. They're all food for the microbes. So yes, feed the microbes, but don't forget to make sure all the microbial types you need are there too.
Biodiversity, right? That's always the goal when we're talking about this. Absolutely. Technical question for you. Optimal carbon to nitrogen ratio, 20:1. Is that correct?
I've seen different studies, maybe 24:1, and I've seen others that are going to be lower than that, closer to 16:1. I don't know the right answer. I can tell you in that 20 to 24:1 ratio, it's a nice mix of having enough nitrogen that the microbes aren't going to tie it up. But because remember, they're going to burn, even fungi and protozoa are going to burn subcarbon off as CO2. So we don't need to capture all of it. They can release it. So that seems to be a nice ratio where we really get good mineralization or release of nutrients and support microbial growth.
Great. Appreciate this. Last question, and thank you everybody for attending. We'll wrap up right after this question and say thanks to Dave here. But this one, I love this question. I wanted to end on it because it really gets down to working with nature. If you're testing your soil for a number of different properties, including microbial biomass, pH, temperature, moisture content, porosity, et cetera, and everything indicates you have healthy soil, how do you know if you have pathogens in it?
That's a good question. So you can do things like, and there are companies out there that are giving services that will identify the pathogens for you. Generally, it's going to be by DNA fingerprinting, would be the best way to do it. An amazing thing, though, is if you look at the science literature, you'll find a term called suppressive soils. And so these are soils where you can have pathogens, they're present, but because you have so much beneficial biology, they don't ever cause a disease. So when your soils are healthy and you've got all the beneficial biology, again with diversity, it's okay if you've got some pathogens. Because remember, microbes fight each other for space and resources. And the good ones are going to exclude the bad ones. When doing field tests and disease study, we have to monitor that field the year before to make sure the disease pressure normally will go up. Because if on its own it doesn't, and this includes nematodes, then we may be working on a suppressive soil, which will screw our results. So it's a great question, but it's also, part of the reward of having really good soil, which is a marathon. It's not a sprint, but it's rewarding in the end.
We need the bad guys to bring out the good guys, right? So we did get a last question, and I thought it's another good one to wrap up on. Do you have any books or references around this subject, especially specifically testing or measuring or improving your microbial life in your soil?
There are a lot of different resources out there that I think are really good. One of the best documents is from the USDA. And I don't remember the reference off the top of my head. If you bear with me, I think it's in one of my slides. If not, I can send it if you guys want to post it. It's easy to read. It's long, but it's in language that I think most people can understand that really goes into a lot of the things I've talked about. Yeah,
I think, Dave, if you want to go ahead and send that along to us, we'll go ahead and put the link on the bottom of the YouTube video when we get this webinar posted and provide that as a resource. So if you find that, when we post that out there, you can go ahead and click through and get the more information. And you'll find links to their website too on there, and links to our websites as well. Thank you to everybody today for attending. Thanks to Holganix and Dave Stark for your presentations today. Again, you can learn more at their website at holganix.com. If you enjoyed this presentation, we have 3 in-person events going on this year, and we'd love to see you at one or all of those, including a couple in California and Wisconsin coming up in August, and our big conference in Kentucky in December. We'll have more in-depth workshops like this available, and you can learn about those. and more at acresusa.com. Again, thanks to Holganix, thanks to everybody attending today, and thanks to Dave Stark for your presentation. Best of luck and have a great rest of your day.
Thanks, everyone. Appreciate it.